Waduh, ada apa gugur loh.
Ladies and gentlemen, guest lecture Biotechnology Approach in Plant Development will start soon. Om Swastiastu. Assalamualaikum warahmatullahi wabarakatuh. Shalom, Namo Buddhaya. The Honorable Dean of Faculty of Agriculture Udayana University, Professor Dr. Insinyur Inyuman Gede Ustriana MM, the Honorable Vice Deans of Faculty of Agriculture Udayana University, Dr. Insinyur Niluh Kartini MS, Profesor Dr. Igusti Agung Ayu Ambarawati MEC PhD, and Dr. Insinyur Iwayan Diare MP. The Honorable Invited Speaker, Professor Masaru Ohme Takagi from National Cheng Kung University, Taiwan. I apologize if there's any mispronunciation. Professor Ming Ser Chen from Academia Sinica Agricultural Biotechnology Research Center, Biotechnology Center in Southern Taiwan. I apologize if there's any mispronunciation. And Dr. Dewi Sukma from Institut Pertanian Bogor, University of Indonesia. The Honorable Seminar Moderator for today, Mr. Iputu Sudiarto PhD. The seminar's honored chairperson, Mr. Putu Perdana Kusuma Wiguna MSc. The Honorable Udayana University professors and lecturers, as well as the honorable guests and participants. Let us give thanks to God Almighty because of His grace and guidance, we are healthy and prosperous. Today's event is held by my Master Program of Agroecotechnology, Master Program of Agricultural Biotechnology, and Master Program of Dryland Agriculture, Faculty of Agriculture Udayana University, in collaboration with National Cheng Kung University, Academia Sinica, and Institut Pertanian Bogor University. And we are proud to present guest lecture, Biotechnology Approach in Plant Development. Before we move on to our next agenda, I would like to say welcome to Udayana University, especially for our inspired speakers. My name is Saras, and it is a precious opportunity for me for being your master of ceremony on today's guest lecture. Ladies and gentlemen, I would like to inform you the agenda of today's seminar. To begin with is the opening and followed by group photo. Second is chairman's report. The third is opening speech by the Dean of the Faculty of Agriculture Udayana University, as well as officially opening the seminar. The fourth is the main event, which include the presentation by invited speakers, discussion session, and the last is closing ceremony. Ladies and gentlemen, now we will have a photo session together, and we would like to invite the Dean of the Faculty of Agriculture, Udayana University, moderator, seminar chairman, and invited speakers to come to the stage and to receive a placard from our university, and followed by photo session with the participant in the background. Please come to the stage. Thank you. Thank you very much, and please to return to your seat. Thank you. Ladies and gentlemen, next we welcome the opening remarks from the Dean of the Faculty of Agriculture, Udayana University, as well as officially opening the seminar. To Professor Dr. Insinyur Ilman Gede Ustriana MM, time and place are yours.
Baik, thank you. Om Swastiastu. Assalamualaikum warahmatullahi wabarakatuh. Salam, namo buddhaya, salam sejahtera untuk kita semua. Good morning for all. The honorable invited speaker, Professor Masaru, Professor Chan, Dr. Dewi Sukma, the honorable moderator in committee for today's seminar, professor and lecturer, the invitee and participant. Let us praise God because of His grace. We are in good health and propitious to attend the guest lecture with the title Biotechnology Approach in the Plant Development. Ladies and gentlemen, biotechnology is one of the applied sciences that is growing rapidly following the demand of the current world of globalization with the impact of increasing complex challenges faced by humans. Biotechnology has played a role in producing food crops with higher nutrition content, plants that are resistant to pest attack, and plants that produce beneficial compounds so that they can be used as herbal medicinal plant. But in additional, the application of biotechnology in agriculture can assist in accelerating seed production, improving plant properties, and producing new type of plant. All that can be produced by means of genetic engineering and tissue culture. Other biotechnology application can be used to create sustainable agriculture by reducing agriculture dependent on chemical. The application of this biotechnology can be in the form of composting, biofertilizer, pesticide, bioherbicide, and bioinsecticide. Various kinds of biotechnology research continue to be carried out to find agricultural products that can increase production or can be a solution to other agricultural problems. Ladies and gentlemen, as stated by the chairman of the committee, this seminar was not only attended by participants from Bali, but also from outside of Bali. Hopefully, this seminar will open us more about the importance of biotechnology and human life. To the seminar participant, I wish you a happy seminar. Hopefully, the knowledge compared with be useful for us, for all of us. My gratitude goes to the coordinator of Master Program of Agricultural Technology, the coordinator of Master Program of Agricultural Biotechnology, and the coordinator of Master Program of Dryland Agriculture, Faculty of Agriculture Udayana University, as well as the National Chen Kung University, Academia Sinica, and IPB University as an integrated part of this seminar. Also the committee who have worked hard to make this seminar possible. Finally, I officially open the guest lecture with the title Biotechnology Approach and Plant Development. Thank you very much. Om Santi 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 Om. Wassalamualaikum warahmatullahi wabarakatuh. Selamat pagi.
Thank you for Professor Dr. Insinyur Inyoman Gede Usrana MM for officially opening the seminar. And now you are free to leave the seminar room if you have another agendas. The chairman of the committee's delivery report is next on our agenda. So Mr. Putu Perdana Kusuma Wijuna, MSc, time and place are yours.
Yes. Good morning, ladies and gentlemen. I would like to deliver some report of this um, guest lecture or this seminar. So I would like to thank you, to the professors who are able to come, Ibu Dewi, and I would like to say welcome to Bali. So we have this agenda for today, October 7, 2022, in this meeting room in Udayana University Faculty of Agriculture. We also have some of participants are available to join us online via Webex. I have to say that most of our students are using Webex today because the schedule of the for today's is kind of bit for this day. So I would like to thank you very much for the committee who are able to deliver the seminar to all the participants, the students, the professors. We are glad to say that we have more than 40%, 30 to 40% here available for us offline in this building. Thank you very much for your coming. We have more than 30 people who are available for us online via Webex. And hopefully you can enjoy this guest lecture, and hopefully the knowledge that's shared today will be beneficial for us. Thank you very much. So I will keep my speech short so we have more time to discussions later. Thank you very much. Have a nice seminar. Thank you.
Thank you very much, Mr. Wiguna, for the report. And now for the next agenda, we'll be guided by our moderator, Iputu Sudiarta PhD. But to begin with, I will read the brief resume of Putu Sudiarta PhD. Iputu Sudiarta earned a PhD degree in applied biology from Shinshu University of Japan in 2011, and he has been a lecturer in the Faculty of Agriculture Udayana University since 2005. He has written more than 25 publications in peer-reviewed scientific journals and international conferences, with 5 articles indexed by Scopus with an H index 5. Pest and disease management of cabbage using Trichoderma sp and Bacillus thuringiensis in Bali Village, Baturiti District, and Taban Regency, Bali Province, with Clemson University, USA, USAID. Knowledge co-creation program for damage assessment method on agricultural insurance with Chiba University of Japan, and many more. So, without any further ado, please welcome Mr. Putu Sudiarta, PhD. Time and place are yours.
Okay, thank you very much for the chance to be a chairman or the moderator for today. So it is a great honor for me because we can together with the special, very special guest lecture. And so before we start, so I would like to briefly introduce for the all lecturer and invite it for the all lecturer to come to the stage. The first, so for this moment, so we have very, very great lecturer from Taiwan and also from the IPB. So the first is Professor Masaru Takagi. Yeah. So Takagi Sensei, so now is current employment in the National Kung University Taiwan. And for the academic background, he is interest for the life science in and agriculture, also in in field of academic expertise for the plant molecular biology, plant biotechnology, biochemistry, and also genetic. And for the education, so for the bachelor is graduate from the Osaka Yuiku University about biology, and for the master for Tokyo University of Fisheries about the algae biology, and for the PhD is from Nagoya University. So he also have the many experience for the work and also the experiment. So this is very very special today because we have the special guest. So I would like to invite Professor to come to this stage. So please. Okay. And the second also very good thing. So now just so before we start, just introduce for the second lecturer, this Professor Ming Tai Chan. So yes, basically he is work in the biotechnology center in southern Taiwan, and so many work in the plant. One of them is about the orchid. Yes, or like the Phalaenopsis. This is a Phalaenopsis also, and so very very great work because have the H index for the scopus about the 38 months, so very huge, and this is very special, and also have the pattern so many, so six about the 6 here. Yes, about the gene and also non-antibiotic selection marker gene, and all about the biotechnology. Also have the honor and the word so many. And also kindly would like to invite the Professor Chan to come to this stage. Just sit here. Okay, so please come. Okay. Yes, this chair for Professor Chan. And also the third is Dr. Dewi Sukma, SPMSE. Is come from Indonesia, of course, so from IPB. So yes, also is expert about the plant biotechnology. Yes, already published in the Scopus and about the seven. Also very great work. And now she is work in Department of the Agriculture and Horticulture IPB, and done by, for the bachelor degree in the Department of Agronomy, Bogor, also in the master and also doctoral. And for the research is about the interest including some aspect, and especially now look at the marigold. Also, she is studying about this one. Okay, so I would like to invite also. So please come for this chair also. So three. Okay. Yes. Yes. Okay. So for the first chance, I will be kindly invite for the Professor Takagi to first chance to present the deliver for the lecturer. Okay. Okay.
Can I? Hello. So I have 40 minutes. So first of all, thank you for the German. Nice to, nice introduction. And selamat pagi. Okay. So I'm a Japanese, and working studying in Japan, and I have a chance to work now I work in Taiwan. Okay, that's me. So the so so next one, please. And also, I have the lots of animation. Okay. So my major is a plant molecular biology, and also the main is I'm studying the functional analysis of the transcription factors. So Arabidopsis as a model plant has 2,000 transcription factors, and we are studying the functional transcription factors. Some people ask me, which transcription factor you are interested in? We are studying all of them. Okay, I'll explain later. All right, please, next one. Okay. Oh no. Oh, it's the animation there. No. Next one. Oh, already. Do we have the animation? It's a PowerPoint. Yes, please. All right. So in this lecture, it's I designed the lecture on site. Okay, and also for the student, mainly for the target is the student. Okay, so who is the student? Okay, so you are too. Okay, so then who is so involved with the agriculture and the plants? Who? Agriculture involved with the agriculture or the plant biology. It's all of them. Okay, okay. So I give a lots of questions to you. You have to answer. Okay. Well, first is please give the animation. Okay, question one. Okay, okay. Do not show the answer. What is the biotechnology? So today we are studying the biotechnology. So all right. So you have first stuck right. What's the biotechnology? All right. So someone can answer that. What's a biotechnology? The answer is already. It's okay. All right. So something like that. Okay, please show the next one. Please show the next one. Okay, something like that. I gave the question, and the student must understand, must reply that. If the answer is wrong, doesn't matter. You know, there's a chance to get the what you don't know. All right. So, but the biotechnology is the kind of the method to increase the productivity and the quality. That the dean already told you. And the most genetic approach is used to genetic engineering. That's important, genetic engineering. And please, the next one. So what is the purpose of the biotechnology in plants? Oh, students gone. So next to you. What's the purpose of the biotechnology? Why we have to use the biotechnology? Why? Okay, please.
To answer with my knowledge, so that the using of biotechnology in plants that we can make the plants better in the quality and quantity side. Thank you.
100% is correct. Okay, but okay, please the next one. Okay, biotechnology is increase the productivity and make a use product and the genetic engineering and please. Okay, so to increase the quality is and the quality and the reduce the cost, you know, something that also biotechnology. Okay, and then next one. That's my big question. For what? It's the answer is here. For what? Why do we use the biotechnology? Okay, please. It's the final the purpose is to increase the quality. Means a kind of the biotechnology is a kind of profit, you know. So finally, it's connected to the profit. So I'm scientist. People say, "Oh, Masaru, you want money or something like that." It's a true. Okay, it's biotechnology is must connect to the company or some agricultural business, you know. And then please, just for the tool for the application, and then for the make the good tool, we have to do the basic science to support to make the tool. Okay, then the basic science is most important for us. Okay? To do the good biotechnology and to make the profit. And just a little bit the next one, please. The doctor in science and too much of the papers. Okay, I did. Oh, this is the next one. Oh, okay. What's the previous one? I found it 40 minutes. Previous. Oh, this one. So okay, in science, a student, please listen. Okay, speaking out is your idea. It's very important. I am Asian. I know that. It's shy. Do not talk. But if I ask, so you can answer. All right. So but American different. Oh, I want to talk something like that. Asian. So we should not shame. Okay, we should not shame the wrong. If you give the answer to the wrong answer, it's good time you can find. I didn't know that. All right. So please think about your idea. Speak out. It's very important. All right. And then please next one. So I want to talk about the literary doctrine. It's too much to go. Okay. Always think about the final goal. If you professor talk to you, okay, to do this experiment. Okay, you can do the experiment. It's okay. But you have to think about why you have to do this experiment. What's the purpose of the goal? Please. Okay. We was thinking of the strategy to approach, and all right, that's press. Okay. All right. Oh no, sir. All right. So then, if you consider the some final goal, all right. So that's most important, and then you can design the experimental method, okay, and then you can select the method, okay, which way to approach that, all right. So prepare. Okay, that's the always think about the goal. Please next one. Too much deducing. Okay. Consider project and set up the primary goal and design your experiment. Right? And without a strong strategy, so you cannot achieve the science, and also you cannot publish any papers. So always please think about if the professor told you, but do this experiment, you have to think about why I have to do and what's the goal of this project. Always think about that. Then you can set up the some strategy, okay, the making strategy, experimental strategy, and collect the materials samples. Okay, next one, please. Too much document, right? Okay, so just please go on. I don't have many time. Please. Okay, science build up the logic. Please, next one. Yeah, try to consider final goal. Always do that. Please, next one. Too much. Okay, for example, if you do some sports, okay, for the tennis, okay, you have to think about you want to be a tennis player or just you want to enjoy the tennis, okay. So then you can think about some way of the training. So you have professional. So that level is very low, but it's just you want to do the enjoy. Okay, so you don't need to do the hard training, something like that. Okay, now come back to science again, right? So your student, what's plants provide us? We are studying the plants, and we get very lots of benefit from the plants. Okay, please speak out. Try to speak out. All right. So next one, please. Okay, plant provides us not only the essential item, that's okay, oxygen, food, but also, Professor, what does the plant provide us? Next one. That's the plant provides the those items. That's our life more comfortable. The rubbers, the papers, and the medicines, So, and such a plant activity, also the healing, So we grow the lots of the flowers. We cannot eat flowers, but without flowers, it's not our life is very pity. Okay. If you come to improve the plant activities, so we can reduce the carbon dioxide and also the food problem will be over. Okay, please, next one. Okay, the question. Oh, by what plant activity is regulated? Please, next one. Okay, these are the plant activity is what regulated the plant activity. Just speak out.
The activities of the plant is respiration and photosynthesis. So they change the carbohydrate into the oxygen that we have to that we use for breathing.
Okay, so the part of the answer is correct. Okay, so that okay, part is okay, right? So, but you have to think about what the professor want to answer, right? Anyhow, it's also true. Okay, please next one. Please next. The function is regulated by the gene, all right? Gene. Okay. Okay. This plant function. This plant function is regulated by the genes. It's encoded in the genome. All right. So all the plant function is the kind of product of the gene expression. Right. So for the most important for the biotechnology, you have to identify the genes, what's to relate to the phenotype that you want to improve, right? So then okay, so that is important, okay, previous, important to detect the genes that regulate the phenotype. That's the first step to the biotechnology, right? So then, please, next. What's the next one? Oh, okay, yeah, that's okay. Please, this one. Please. Okay. So this is a dates under the oil palm. Oil palm is very famous in Indonesia. You know that, right? Every day you use the oil palm oil. So, okay. So under you know the dates? You know dates. It's some sweet, sweet one. So you know what's the dates under the oil palm is very different. But you know how much the genome is different. Okay, so the dates on the oil palm is a completely different the palm, but the 99% of the genome is identical. Okay, just only the few genes is different. That means that if you can find some difference of so some gene produce the oil. Okay, sugar, they make the synthesis and they produce a lot of the accumulated sugar, and some is go to the oil. So some of they go to the dates. It's the difference. So if you find some key factor, key genes in the between these genes, you can modify this palm to the oil palm. Right? It's possible. That means that for the biotechnology to identify the genes most important. Please, next one. Yeah, for biotechnology approach, what we need is identify genes that regulate the trait. Okay, so this is the cyclamen. Cyclamen has 5 petals, but just by modifying the one gene, we can make the rows, the convert to the rows. Just only one gene, so we modify that. Okay, today I don't have time. I don't have many times, so just to show that biotechnology can do this, okay? Just to introduce that. Okay. So then, how to identify the gene? Oh, okay, identify gene. Awesome. Okay, so you already showed the answer. Okay, it's okay. We don't have time to do that. Okay, how to identify genes? Each trait. It's please next one. Okay, so loss of function analysis is the most critical. And so I'll show what is loss of function, please. Okay. For example, just the one gene mutant. So the difference you can see difference. This is how the stamen, but this one mutant do not have the stamen. And what's the difference with the middle one? What's the difference? How do you think? What the gene is mutated? This is the right side is mutated. Yes, okay, that's correct. Okay, she said the difference of the color is a difference. The wild type has accumulated the unsigning, but this mutant do not have the unsigning. That means that we mutated the genes that they produce the unsigning. So that means that we have the color gene of the unsigning. Okay, next one, please. This is the okay. This is the loss of the root hair. Just mutated. The plant produce a loss of the root hair. Mutation is not only the loss of function, but sometimes increase the some function. All right. So please next one. Okay. So going. How to play the knockout lines? Okay. So please next one, please. All right. So and we have to find some genes. We have to make the knockout lines. We have to kill the genes, each of genes, right? So how to do that? In plants, I just explained. So EMS, ion beam, and T-DNA tag line. Also, the natural variation of is very important to find the genes or knockout lines. you know the difference of knockout and knockdown. Knockout and knockdown. You know the difference? Oh, I can translate to English. What's the difference? Okay. Okay. So okay. So knockdown is just knockout is just kill the genes. It's a genome. But knockdown is after the transcription, suppress the transcription by the antisense RNA, something like that. Okay, please go on. Okay, so summary for the so far that we talked that to make the plant biology to make a modified manipulation of the plant trait using biotechnology. It is important to identify the genes that to regulate the phenotype, and we have to think about how to identify the genes. Okay, the plant trait is a loss of function analysis. Most of the good approach. Please go next one. All right, so all right, I skip this one. Okay, please next. Going. Oh, okay. Next one, please. Okay, so this is a machine. It was an ion beam. It's a press the added the ion beam and destroy the gene to make a mutant. You know. Some it's advantageous. We can use some materials, big materials. So it's a very useful for the change the color of the flower, something like that. This is the gamma field. We are second teravector, 88 teravector. And it mutated. Right? And try to find out some strange lines and try to fix the genes. Okay, but please. But it usually takes a long time. Only in the vegetable, the 5 to 6 hours, 6 years, and also the fruits it takes 20 years. So if you started the experiment, so your son or some grandson can establish this fruits, for example, the line. You know. But the genetic engineering can shorten this time or much short. Okay, please. All right. So next, okay. So also the loss of function is something mutated gene, but also gain of function is also useful. And this is the purple tomato is overexpression of the genes of the unsigning. So they accumulate the loss of the unsigning. It's come to the purple. Sometimes if you get the loss of the unsigning, is maybe they're helpful, right? And also next one, please. Just introduce. This is a tDNA tagline. As the agrobacterium can transfer the genome, transfer the genome to the genome. Okay, so then around the 20 kilobases tDNA is inserted to any randomly portioned in the genome. So in Arabidopsis, has 26,000 genes. Almost 98% of the Arabidopsis genes is knocked out already. Right, please, next one. This is in a website you can find some knockout line in Ohio. So if you are interested in this gene, you can easily find out the knockout lines, and you can request and get the knockout line in Arabidopsis. Okay, and then next one. That's a question. So what's the genes? Please guess. Okay. First, we made a mutation. I'll get to the something mutant phenotype. You have to think about what the genes is mutated, right? All right. So you escape here. Last one. Oh, okay. 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 First one. It's the first one is a not easy question. First, the second is just easy. The red color is disappeared. So that means the ancestral genes maybe involved that. Okay, first one, we mutated. It's a flower, leaf increase. All right. So how to think? What's the genes is mutated? It's not easy question. Okay, I'll give the answer. Okay, plants just grow this way, and gradually. But it's called apical dominance is regulated, But the genes that regulate the apical dominance is gone, all right? So that means the parents press that, but the parents go out, it's the children and make the lots of the leaf, something like that. And you know what is the D? How to think? What a D? D is a cup. Okay, cotyledon is the two leaves, but it's down to the cup. What's the gene is mutated? How do you think that? Okay, so answer is there's some scissors. Okay, scissors to usually cotyledon is usually it's a fused one, but to make the cotyledon there's a scissors. Okay, we mutated the scissors, but then so that the cotyledon fused cannot separate that, something like that. And this E and F is the same genes is mutated as a no stamen. It's only the female organ. It's named the superwoman. Okay, something like that. Okay, so how much I have? Oh, I have still 20 minutes. Okay, the next one, please. So, but there's so many genes that we have to analyze that. So rice has at least more than 53,000 genes. We have to study the one by one. It takes time, but is there any effective pathway? That's why I'm studying the transcription factors, but I skip the detail about this. Okay, so the another question is the biotechnology. Biotechnology, the purpose is how to increase the qualities and the quantities, and how to reduce the cost. The cost means the fertilizers and the labor. Biotechnology, you can think about what the labor we can reduce the biotechnology, something like that. And also, most important, how to increase the profit, final profit. Okay. And please, this question: what's the major plants in Indonesia? The crop, vegetable, flowers, trees, and rice, fruits, orchid. So please think about how to increase the such important plant in Indonesia, something like that. Right. So what's the question? Oh, that's the same one, right? Oh. Okay, what's the major plant in Indonesia? You are Indonesian. You have done. What's the major plant in Indonesia?
Rice, sir.
Rice is very important. And then
Palm.
what's a palm? Lots of palm there. Lots of palm.
Coffee.
Great. Oil palm produces a huge money, right? And something like that. Please, next one. Yeah, okay, please next. And then, okay, so then to increase the some plant rate, this is very important question. what is a plant, and what's the different from the animals? Okay, so this is the some lecture, and you don't need to write down here, but okay, you student must answer what's there are the 5 useful traits different from the animals. Okay,
Photosynthesis.
100% correct.
When I couldn't hear.
Difference.
On plant they have chloroplast on their cell.
It's a chloroplast. It's photosynthesis. All right. So okay, who is the student? Oh, here's two students here again. Okay. Yeah, please press on. Please press everything. All right. So this 5 trait is not exist in animals. Okay. For example, there's so many lines. Photosynthesis is only the plant. Right? If you synthesize the chloroplast, we can get the carbohydrate, but we cannot do that. Okay. So photosynthesis is provide not only the essential items such as the oxygen and food, and also it's a many items that produce that. And the totipotency is the only plant can reproduce from the single cell. Okay. But animal cannot do that. If you pick up the liver, or liver here, all right, pick a liver, and if you cultivate, only liver can you can produce. You can produce the baby or something like that. Only the egg cell can be the baby. But the implant, if you pick up the some cells and cultivate, you can regenerate it. Okay. And also the okay, sessile organism. That's very different from the animals. It's sesame that cannot move. Okay, plant cannot move. It's algae can move that, but the plant cannot move. That's the big trait. Mean the a plant has a lot of the stress resistance system. Okay, a plant cannot. Animal if too much sunshine, we can escape to the shade. You know, or if the rain can have the umbrella, but plant cannot do that, right? So the plant has developed the lots of the some protection system. Okay. And the other is also the directly involved in the profit. Plant produce a lot of metabolites, you know, and around more than 2 million, no, 20,000, 200,000 the metabolites are already identified. So then, and next one, please. So which traits should be manipulated to increase the productivity and quality and commercial value? So first, the most the easy is approach to this one, stress response. Please next one. Okay. So they developed the all right. I skip this one. Yeah, how to increase the quality and the productivity yield. Okay, question. And next one. Answer. So increase the stress tolerance. Okay, that's the first approach of the biotechnology, plant biotechnology. It's an easy way to do that, right? So if we can convert the tolerance to the various environmental stress, the productivity will certainly increase. And then this is the next question. What's the plant that is sacrificed? What's the stress for the? I know that this is stress for you. Okay, my question. I understand, but what's the plant? Yeah, this is stress. Yeah, can I answer that? What's the stress? Okay, you don't know the plant do not have the stress. You said that oil palm. Okay, someone said the rice is important, all right? So rice productivity in Indonesia is very variety. So Bali is some nice country to produce lots of rice, but the job Jakarta is a very poor productivity of rice. You know why? It's a different of it's a soil. What's the difference?
Quality, minerals.
Okay. So okay, I show that something like that. What's the stress? Please next one. Yeah, please. Okay. So stress, environmental stress is temperatures. You know, too much hot. You can produce some apples. Only import. You have to import that. And also the high salt stress and physical stress, strong wind and rain and the sunshine, or soil. That's the Indonesia. As acid soil is the south area has a volcano, and also lots of the covered by the acid soil. So the plant is very especially rice do not like the acid soil because acid soil produce release lots of aluminum. Aluminum is inhibited the root development, right? So that's directly involved in the productivity. So if you can remove such a cause or such a problem, okay, you can increase the productivity and get just increase the profit. That's the biotechnology. Okay, so and also please, and also the nutrient. Nutrient is also more important. Okay, low nitrogen, the low phosphate, and okay, and also virus. All right, pathogens. It's we have for the long history, the people fight with the pathogens, and they developed the herbicide, the insecticide. Okay, lots of the chemicals we developed. So we can produce increase the productivity, but also at the same time, it's harm to our body. The herbicide is not so a big problem, but the insecticide, really the bad chemicals are sometimes mutated in a human being. All right, that's a bit, sometimes affect the baby, something like that. All right, so all right, so just okay. So there's some summary of that's a plant house storage. Okay, not this lecture. Okay, all right, something like that. Just follow that, and then next one, and also acid soil. It's a red part is the area of acid soil. Indonesia is completely red. So that's directly involved in involved in the productivity. And Brazil has also acid soil, but they develop, they change their soil condition, and now Brazil is the #1 country for the soybean production. That's it will be possible to do that in Indonesia too. All right. So how much do I have? Oh, only 10 seconds. I have to closing. All right. Just with 5 minutes, with 5 minutes we close. Okay, please next one. So, okay. Oh, sorry. It's okay. It's okay. Okay. Yes. All right. So I'm finishing it with 5 minutes. Okay. I just show that just one modification of the genes, we can produce the acid soil tolerance line. Okay. And then please show the next one. Next one. Okay. So and also fertilizer. And the next one, please. Okay. So I skip this one. Next one, please. Next one, please. Oh, okay, please. Okay. This is wild type, and this is the 100 times we reduce the nutrient N. Okay. So, and also we have succeeded to run to the tolerance of low nitrogen. Okay. So you said the oil palm is a very important food in Indonesia. Now, how much the people spend their fertilizer for the oil palm production? You know that? More than 30% of the total cost is fertilizer in oil palm. So if you can reduce the nitrogen fertilizer in oil palm, so there is a very, very big contribution for the environment or also the company. Something like that. Okay, please. I skipped this hospital. Please. Okay, go, please. Okay, please. Please press that, and also press that. And also press that. This is hospital tolerance. Just only modified one gene. We can show the tolerance to the low hospital. And then next, please. And this is heat stress, and the wild type cannot survive that. One modified just one gene, so we can make the heat tolerance just in Arabidopsis. Okay, please next one. This is salt tolerance. It's a seawater is 600 millimolar. 400 millimolar. That's very very salty. But just modified one gene, so we can make the some tolerance line to the salt. Okay, please next one. And okay, please next one. It's a nematoda is also serious problem that. Okay, and the next one. And this is a striga in South Africa. It's a serious problem for the some. It's a weed of the witch. They say so. It's infected the rice or corn and feed the nutrient of the plants and that completely kill that. Okay, next one. Okay, next one. All right. Okay, just keep. So also okay, please. Just one modified the okay, please next one. Please. Just one modified the one genes. We can increase the oil content. Arachidopsis has a 34% is average, but just modified one genes. We can increase more than 20% of the oil. That's the some change the metabolic flow. That's in biotechnology can do that. Next one, please. And also, if we can apply this gene to the oil palm seed, we can increase the oil production. And also, next one. And this is also the important item, incense. So I'm Buddhist, so we use a lot of but this is produced by the plant gene. Okay. All right. So it's starting the infection of the some virus, but we can. This is a gene is in the genome. So if we can find the necessary genes and modify it, we can produce a lot of the incense material, right? And its commercial value is very, very high to do that. Okay, next one. I'm closing soon. Okay, this morphology, we can change the just one gene, so we can modify the morning glory. This one, right? And also this one is it's a morning glory, it's double flowers. You have ever seen this one? Morning glory can do that, just modify the one gene, right? And next one, we apply to this gene to the cyclamen. Okay, please go. And cyclamen increase the presence. More than press. Okay, 200, 300 petals, and maintain the 3 months, something like that. Okay, and skip. Okay, please skip. I don't have time. Going, please. This is a tomato. Okay, so final question. What's the difference of the tomato?
Less seed.
No, not less seed. No seed. Okay, no seed. Okay, so just by modifying the one gene, we can produce the fruits without fertilization. So it's a boy. You don't need that. Just male can produce the fruits. All right. So that means that so it's a kind of contribute to the oil palm production. We can produce the oil palm meat. With no seed. Okay, to increase the fruits. Also, if there's some now I don't know Indonesia, but in Japan, there's sometimes the weather is so changed. They fail to produce the stamens. Okay, so please next one. Okay, please next one. Next one. Okay, this one. Okay, please present. Okay, final. This is last slide. Okay, so please think about the strategy to increase the quality and quantity by using the by modifying genes. For example, oil palm. Oil palm is from 1986. Productivity per hectare didn't change, right? The kind of saturation. So, but if you increase the productivity of oil palm, so two possibilities. We do not need to expand the oil palm plantation. Oil palm plantation is I don't know, but many people kind of hesitate, so because the oil pollution, there's a producer huge pollution about that, and also the lavas, and also the lava is very important in Indonesia, right? But the productivity is the same because they do the lots of the labor, the cut every day, and you need a huge area. If you how to reduce the some labor, and also rice. Okay, so rice is a very important product in Indonesia. So there's two possibilities: just increase the productivity and also increase the quality. But if you have the some a kind of so-called low quality rice and the price is cheaper, farmer is not happy. But if you can increase the high protein, it's a kind of the not tasty rice. Human does not eat that rice. It's bad tasty. But if you increase the protein, okay, it's very useful and for the feed. Everyone in the world, people want to feed for the cows and some chicken, something like that. Okay, and also okay, that's a please. As a crop, probably Professor will, David will talk about that. Okay. So the next one, please. Next one, please. Next one, please. Our next one, please. Okay, thank you for the listening. It's over time.
Okay, so thank you very much for Taki Sensei. So arigatou gozaimasu. Before we have discussion, so we will be continue first for the okay. Yes. Okay. So next, before we move to the Professor Chan to delivery for the lecture today. So I think it's very before we start. Professor Takagi is very nice lecturer. So because can make the student stress. Okay, this is important thing. Okay, thank you. Okay. Okay, time is yours for Professor. Okay.
Could you hear me? Okay. I will not ask the question, so don't escape. Yeah, the student almost escaped. Okay, I'm Mintai. So it is very easier to give a talk after the Takagi sensei, because Takagi sensei just give his philosophy, yes, philosophy question for all of you, and I may give you some practice experiment. Experiments. So okay, today I will give the talk is the cell manipulation of the plant improvements through the genetic transformation. However, my main things may be describe the genetic transformation. So if anyone are doing performing the transformation, I think that might be useful for your research. Okay, next slide. So because we already know that the genome sequence data are available for many important crops, plants. For example, like the tree, corn, rice, Arabidopsis, soybean, brassica, and so on. A lot of. So next. So that's why I have to say that genetic modification is another breeding methods. Although the traditional breeding, it may spend long time and labors to reach the purpose. So the genetic modification is just another tool to get earlier to get our favorites crops. For example, this one is for the when we can find easier to in the wild tomato cultivar tomato species. It may yield poor yields, but they can provide us disease resistance. So then, if we can identify some genes resistant disease, then we can isolate this one, then to transfer to the cultivar, tomato cultivar. Then that tomato cultivar to on the disease resistant characteristics. Okay, next. So the whole procedure is just as like Takagi Sensei already mentioned, as any gene you may be is favored to us that we can clone. So the resource might be from the human, even the human, or microbial, or plant, and some virus. So the resource of the gene we can clone, then we can call it the genes of the interest. Then we isolate this interest by molecular biology tool. So now at this moment, I think it's very easier to clone a gene because due to it's easier, you can use the design primer to directly amplify the using the RT-PCR to clone the cDNA. So now it's very easier to escape the genome intron and others. So then after the cloning of this gene, we can insert into the vector, then using the vector to transform to the crops. Next. So as compared to the traditional breeding method and genetic molecular genetic engineering, you can see that it's almost the same. Except that the GM crops, that means genetic modification, you can isolate a gene come from any resource, and you don't need to analyze the genetic resource. You can directly transform to the crops, and also the plants can be propagated vegetatively. So that might be spend less time and labor as compared to the traditional or now we call it molecular breeding, because you have to find in the field, you have to find a deserved threat in the population. Then you have to identify the potential genome. Okay, then you have to cross. Then you search. You can find that the useful characters in the fields. Okay, next. So because this is the guest lecture, so I may use the more practice one for you. So the precaution for the success transformation might be we have to look for at least the following factor. First one is to build the plant tissue culture system. If you are doing the tissue culture, I hope you are not upset. Because you may think that tissue culture is very easy, but I think the tissue culture is art. Okay, because you have to know when you have to transfer the material to another medium. Okay, yeah. So next slide. So because so now if you can establish the tissue culture, the whole procedure, then you obtain a regeneration plan. That means you already get the first step for to obtain the transgenic plant. Okay, so any material in the tissue culture procedure that may be useful for the plant transformation. Next. So okay, so because the regenerative transgenic plant from the transform cell is very difficult. Okay, So if you already obtain the regeneration system, that means you already get the first step to obtain your transgenic plant. Okay. Next slide. The second step is to clone your favorite genes, like Take Sensei mentions that any favorite genes might be disease resistant, virus resistant, temperature tolerance, okay, salt tolerance, or drug. Next. So before we go down to for the following the procedure, then we may review again the gene structure of the eukaryotic cells. We need a promoter region in the right side. So it's very similar like the promoter region. Like you have the Tata box and the Keta box and other regulation, upstream regulation region for a specific tissue expression. Okay. Then you may need the 5' untranslated region and also 3' untranslated region. Now, because that we already easier to using the obtain the cDNA without using the RT-PCR, so we can easier to obtain the full length cDNA. Now, okay, so you just design your promoter and your favorite gene and the terminator. Then you can do another transformation. Next. So before you do the transformation, you need a one vector. For example, that might be the antibiotic selection genes. We always use that neomycin phosphotransferase for selection in the plants. Of course, we also need to the bacterial selection marker for the selection your Agrobacteria or others. Then in this slide, you can see that the DNA from your interesting to integrate into the vector. Then sometimes we will use the overexpression to drive the marker genes, okay, or the antibiotic selection marker. The next slide. Okay, so to be expression in the plant cell, a gene has to look like a gene, plant genes. Okay, so that means you have to find that suitable promoter to drive the gene expression. So now we always use the certified cauliflower mosaic virus certified promoter to be the overexpression to the antibiotics genes. So you can see that we still need to the promoter, like 5' untranslated region, open reading frame, 3'. Okay. And so that is the whole construct you have to do before you do the transformation. Okay, next. Then to achieve the high level of the production, sometimes we may have to use the useful promoter. Like the constitutive promoter, we always use the cauliflower mosaic virus certified promoter. This is suitable for expression foraging in the die cut. So especially only in die cut. So because in the monocast, the certified promoter, that may be expression very low expression. So sometimes you may get the low expression of your targets. So if you want to use in a monocotyledonous plants, you may use in the maize ubiquitin promoter or rice actin promoter. That might be more useful for the constitutive expression. Okay. So that might be the difference between the dicotyledons and the monocotyledons. So next slide. And to another one is some specific tissue, specific expression promoter, like glutamine to expression the C specific, then alpha aminos in the allelon or for the rice, the protecting in the potato, in the tuber, or rubisco small subunit, especially in the green color. Okay. And sometimes we will enhance the SRS. It's the enhancer just behind the promoter. Okay. To enhance to expression the genes. Okay. And if you want to increase the protein level, we also will add the signaling, reading signaling like a KDL in the end of the amino acid. Okay. Next. So the third step is to choose the parameter material. So that is very important in the tissue culture. Okay. Because any material is easier to regeneration. That might be the good material for transformation. Okay, next. For example, the rice, you may use the embryo-derived callus. So because callus, it can propagate very easier and propagate very faster. So that means when you're using the agrobacterial transformation, that will be the foreign gene might be easier to integrate to your genome. Okay, so every the step, the important step for the transformation is when you choose the easier regeneration material, that you may obtain easier to obtain the putative transgenic plant. So, for example, like the rice, the oki is also like the protoconn-like body. The protoconn-like body is the like when you obtain the protoconn, because the oki did not have the seed, so they will germinate like a protoconn. After the you can cut the protoconn, then to form the protoconn-like body. That would, because they can propagate very easier. So that would be the good material for the transformation. And a tomato, that might be the cotyledon or the young leaf. The soybean also is cotyledon nub. Okay, others. So every fast growth cell or tissue would be your easier obtain or your most important material for transformation. So keep in mind. So I think that marigold might be, you can use the callus. Right, okay. Next. The next step is to design the transformation methods. Okay, next. So because a lot of the transformation methods already development, so let's introduce some important or common use the method. Okay, the first one is the Jingang. Next, I think this is the physical transformation tool for everyone. However, this equipment is very expensive. It's about, how to calculate, 50 no, 500, how to calculate, 1 million NT dollars. 1 million NT dollars plus the 500, okay, for only once. So not every facility can provide this material. However, it is very useful for the transient test. So next slide. So the principle of this, the Jingang, is just using the gas to push the microcarrier, the DNA. The DNA combined with the gold particle. So then the gas to pass down, then they will spray. So the tissue will be put over here, target tissue. So then the gene accompanied with the gold will shoot into the cell. So not only the plant cell can use this system, but also the animal cell, animal system, animal cell, okay, cancer cell, and you can do also can do this one. Okay, next. So another one is handheld Jingang. This is another, the portable Jingangs. You can do in the field for the transformation. Okay, let's see, next slide. You can see that you can do your genes to shoot every plant culture in the field. And also you can test each construction in each leaf. You can see that we cover the, and then we label which gene we may test. Okay. So the second one is biological tool. It is the very easier. We can call it this poor man transformation tool. Poor man, because why is poor man? Because we only need the agrobacterial. Next. So the agrobacterial can modify it just because long story shows because we can modify the TI plasmid because the TI plasmid is very big, difficult to manipulation. So now we can miniaturize the vector. We name it. We call it binary vector. It's around 10 kilobase vector. So it's very easier to clone. And accompanied with your regeneration of the your regeneration system, you have the chance to obtain transgenic plant. Okay. So let me brief the procedure. That's in the field, the original agrobacterial can transfer the tDNA into the after the wounding site. Then tDNA can integrate into the plant chromosome DNA. Okay. However, before we know that the tDNA, the structure, we don't know why the agrobacterial infection, after agrobacterial infection, they will show the ground gold. Now we already know that because the tDNA containing a lot of the plant hormone gene synthesized genes, including the auxin synthesized and the cytokinin synthesized. So they will let the plant cell to produce a lot of auxin and the cytokinin. So then you can see the in the field, and you can find a lot of ground gold. But I think that it's not easier to find agrobacterial in the Indonesia soil because that agrobacterial is very sensitive to the temperature. Okay. Maybe you can find the agrobacterial strain, some microbacterial strain in a mountain, maybe. So because why? Because agrobacterial after the over the 28 degrees, that the tDNA TI plasmid will destroy automatically. Okay, so that's why the agrobacterial you have to easier to find in the high temperature, low temperature low temperature areas. Okay, next slide. Okay, I have one exact sample that's for the transformation. That is the Arabidopsis. Arabidopsis is the model system. Maybe I can say that the Brassica species can be used the flowering deep transformation. That means that we're using the agrobacterial to flowering deep the Brassica flowers. Then we can obtain the putative transformants. You may see some paper describe they use in the flowering dip in the maize or oki or rice. My advice is don't believe that. Okay, because we already test a lot of the flowering dip in the many crops. So you may find that those papers only published in large journal. Okay. So if you do the Arabidopsis or Brassica, then you can do the flowering dip method for agrobacterial. But if you do the another crops like the marigold or maize or oil palm, that you may use in the tissue culture system. Okay. Next. So next is electroporation. Okay. why I have to introduce this one? Let me see. Let me talking about, because the print on the cell wall, secondary cell wall. So we have to use in the enzyme to digest the cell wall to form the protoplast. Then we can isolate the protoplast, then using the PEG or the electroporation, okay, to obtain to let the foreign gene to into the protoplast. Then we culture in the medium containing the antibiotic. Then we can obtain the putative transformants. Next slide. Next slide. So this is the electroporation, but I think this one is also very expensive one. But if you want to do that, you may also use in a PEG and calcium together. Then that can also can do easier to do the transient inspiration. Okay, next slide. That is the electroporation principle that you will use the high voltage to let the cell wall, the protoplast to open a lot of small pore, let the DNA into the cells. Okay, next. Why I want to introduce this technique, because that's next. Because recently a lot of the CRISPR technology has been development, and also because the CRISPR trend we may think they are not the GM modification because they may not contain the antibiotic selection genes. So if you want to use the protoplast, just using a CRISPR vector, you don't need to select the antibiotic. So you can direct using a CRISPR vector to introduce in the protoplast. Then using the T1 digestion to using PCR to obtain the putative one, and also the deep sequence to obtain the CRISPR crops. Okay, next. Then the next step is to select your putative transformant. Okay, next. So before you do the select the putative one, my advice is just test the antibiotic concentration. Why? Because you use the material maybe not the typical identical to the paper description or paper used. So you may test your own material before you do the antibiotic selection. For example, this one, we're using the Oncidium. This is another OK, Oncidium cherry baby. So this cultivar, we call it OM8. You can see that it's about 10 microgram per microliter hygromycin. They cannot survive after 2 weeks. However, if we're using another Oncidium, OK, we call it this is cherry baby. Another one is sweet sugar. Sweet sugar, they can tolerance to 25 microgram per microliter. So that means, although they are very similar species, but their tolerance, the antibiotic concentration is totally different. Okay, so next. So after you test the antibiotic, so then you can see that we use in the OK transformation system. We use the P2B, the co-culture, and to selection and to obtain the putative one. And also the rice transformation is also. Okay. And next step is to detect your gene expression of favorite genes or selection marker. So next. So this is before our work. This is a previous work. Previously, we always use the gas staining for the transformant, but now we almost discuss this system because we may easier to find the gas-like enzyme in some crops. For example, like oki is another one. Oki, especially like oki, Phalaenopsis oki. Yeah. But however, that southern blast is the evidence to prove your florigene has been integrated. Now you can easily use a genomic PCR, RT-PCR. Next slide. Then to see that using a northern blot to see the hygromycin antibiotic selection system, genes, the Gus genes, marker genes, and also the Gus for the Western blot for the protein expression. Yeah. Next. So that's after that, then you can transform your favorite gene, like the Takai Sensei mentioned. Okay, so for example, this one is our transcription factor, AP2, to enhance the chewing tolerance. Our system is the overexpression that. His system is repress that. Okay, so and also we find some tomato ice-like genes is sensitive, is very enhanced chewing and the water deficient tolerance. Next slide. Then you can evaluate the whole procedure. Yeah, no matter you use the agrobacterial method or a gene gun method, so you have to choose the easily propagate material. Then you can through the regeneration system to obtain the transgenic plant, the plant with the nutrient. Next slide. So this is the whole procedure like we established in the OKI system, and we propagate a protocol-like body, then selection after the using the agrobacterial mediated transformation, then selection the protocol-like body on selection meeting, then obtain then the regeneration and culture in the field, greenhouse, and also analysis the foraging. The next. The tool is no matter what genes you may isolate. Maybe you from the bioinformation you can obtain some favorite genes, vitamin or proteomic or metabolomic. So the gene discovery, then you can do the forward genetics or reverse genetics to do the concept, the genome concept proof. Then you may obtain the novel gene to the crop improvement. Next. Okay, so let me, my center. Now we are moving to the Tainan. It's the biotechnology center in South Taiwan. Okay, another place is Nangang. Okay, next slide. Let me give the advertisement for the student. Yeah, we also have the collaboratory program. This means the NCKU, National Chengkong University, and Academic Snica graduate in the translational agriculture. So we need more people to join us. No matter you want to study the master degree or a PhD degree. So we welcome everyone to join us. Next, you can see that the website. Then, okay, thank you. This is one.
Okay, thank you very much, Professor Sun. Now we move to the third lecturer for to Dewi. Yeah. So she studied many about plant and including marigold, gemitir ya in Indonesia or TGT something like this one. So I think it's interesting. Will be more interesting when she will make the merah putih marigold. So like this one. So now time is yours. So please prepare your thing. Okay, thank you.
You.
Thank you for the moderator. Good morning, everybody. Assalamualaikum warahmatullahi wabarakatuh. I'm honored to be here in front of two great professors. So if at the first session the student are nervous, no, my time to be nervous because there is two great professor here, but I have to do this. It's a process of learning for me. So if I may tell something wrong, so my professor will know how to correct me. Thank you, Professor Takagi, Professor Chan, that you always they always guide me to improve my knowledge, my techniques, and also accompany me to guide the students, to transfer the knowledge, the inspiration, the spirit to be better for our generation. So my talk today is biotechnology approach in marigold breeding. Why do I choose this topic? Because we are supported by the Bali government to research these plants. So Bali government is very attracted to these plants to wake up Bali in the agriculture. So okay, our team is led by Professor Muhammad Syukur, Dr. Sarif Aisyah, there are Dr. Deden, Professor Sudarsono, and Professor Dewa from University Udayana, and Prof. Chan from Academia Sinica. Next, please. So as we already know that marigold is famous plants, especially in Bali. There are two famous species, Tagetes erecta and Tagetes patula. These plants are native to Mexico and Central America. In Indonesia, we call it gumitir, maybe in Bali, or gemitir, gumitir, right? And in other areas in Indonesia, like in Java, we call it tai kotok because most of us doesn't like the smell. Right? But no, I already adapt with this smell. No, I like it. So there are many uses of this plant for bedding plants, cut flower, edible flower, medicine, tea, and maybe for spiritual ceremony in Bali. And we just know that market demand for these plants is very high. So these plants have the high economic value. But until now, I see that Bali people only use it in the daily activities for maybe for spiritual matters. But actually, these plants contain many kinds of biochemical compounds that can be used for dye, for medicine, for cosmetics. And yesterday I met a friend from Sultan Ageng Tirtayasa. She already applied many kinds of plants for handicraft, like for textiles, and maybe someday we can invite her to introduce the techniques. So it can be it will be very useful for Bali people to give the value to add the value of the ornamentas. Then next, this is the existing color variation and flower type of marigold for the Tagetes erecta species. So we see it's very good color, but only in ranks of vanilla, yellow, orange, like that. We do not found we did not find the color, the true color in red of Tagetes erecta. Then the other species is Tagetes patula. They only have a mixed red color, maybe red maroon, then mix of yellow and red color. So actually they have very limited color. Next. So we have started to breeding these plants by using 3 approaches. First, by conventional breeding to cross them, to do selfing and to select the progenies, which one is good, which one is not. Then we propagate to be developed as new varieties. Then the second approach that we do is mutation breeding by using colchicine or gamma irradiation or methyl sulfonate. And the third method is by in vitro and molecular breeding and transgenic. The final aims of the research is to find the new varieties of marigolds. Next. And what we have got so far, we found that in conventional breeding, we have the problem to cross these flowers because the flowers is a kind of composite flowers. They have the ray flowers is so this part of the flowers is ray flowers, not yesterday Prof. Chan explained me that this flowers, right? Flowers, not petal, but they are flowers. And the center one is the this flower. So we have the difficulties to cross each other. And the other problems is that we only get the small number of seeds. Then after we cross with the only limited number of seeds that we get, then after we plant, we grow the progenies, we did not get the diverse color. So the color only in the range, red, maroon, or yellow, or orange, or mix of yellow and orange. So next. So these are the some genotypes, some lines that we already got from our research. We can see that the color still in this range, right? Then next. So this is the F3 population. So after selfing 3 times, we got the F3 and F4. So we see the color is still orange or yellow. Next. Also in Tagetes spatula, the second species that we research here, we see the original color here. This is the original color. Then after we treat it with the colchicine, the kind of chemical that can change the genes in plants. Then we see the right side, the color of the flower still in that range. So we cannot get new color. Next, please. This is the result from mutation breeding by using gamma irradiation. So we see quite high variation compared to the previous one, compared to colchicine, because gamma irradiation give a hard effect for the genome. So, but still, the color that we got still red and yellow. And we find some new morphology in this quite different with the wild type, with the original flower. Next. And we also did the mutation breeding by using ethyl methasulfonate on this genotype, the vanilla color of marigold. Then we cannot get the pure white because actually we like to get the pure white and puret of marigold to refer to our flag, national flag. Then, so far, we still have limited number color that can we achieve from our activities. That's why could biotechnology help the marigold breeding to get a more diverse colors? In example, pure white or red white or red, pure red. So if I'm not wrong, Prof. Takagi, why do we need biotechnology? Do you ask it? Why do we need biotechnology? I always nervous is if Prof. Takagi give a lectures because he always will ask the question. Now, now I will answer his question. I will answer your question because by using biotechnology, we can make what we can make what we want. That the morphology or the characters that we never imagined before. Do you believe it? Yes, because Prof. Takagi and Prof. Chan already show it, like the cyclamen, from only a single petal to be more petals. Like so, that's why we need biotechnology to uh, to get the character that we want that cannot be achieved by the conventional breeding. So in biotechnology, we can introduce the genes that come from other genetic pool, maybe from animals, from bacteria, from fungi, even from yes, from different kingdom of the what is that? Different kingdom of ya dari berbagai sumber lah gitu ya. So next, what we plan, what we are doing with biotechnology on marigolds. So if we see the plant biotechnology could be in the scope of cellular to molecular level. Here I highlight the three part that we do know: tissue culture and plant regeneration. Then we do the transcriptomic analysis and genetic transformation on marigold. Next. So this is the step that we do from the tissue culture. We optimize the regeneration method by tissue culture. Then we use the construct that provided by Professor Chan. There are the three constructs that we use. RNEI for TSJ, then in MYB CCD4 to degrade the carotenoid in the flowers, and the other construct is CRISPR to mutate the gene that make the carotenoid in the plant. Next. So until now, we found that for regeneration by direct or indirect organogenesis, we use the plants. We use the explants from leaves and internode, and so far we found that to induce callus, it's better if we use the leaves, in vitro leaves. We can use it to induce callus. Then from the callus, the shoot will be comes out. Then next. So this the figure. If we use the leaves and internodes from the in vitro plants, we will see that most of internode will make roots. while the leaves will make callus. Then from the callus, the shoot will grow. Next. And we improve the efficiency of shoot regeneration by modify the auxin content in the medium. Because if we need more shoots from the callus, we can get more plants. That's why we have to fix the regeneration methods in tissue culture. Next. This is another picture when we use different composition, different concentration of auxin and cytokinin in the medium. Then we already found the optimum concentration to get more shoots. Then after we fix the regeneration methods, this is the requirement to us to do the genetic transformation. Next. So we can get the shoots from the callus. Please next. Now, how can we modify the color of the marigold by biotech? I just mentioned that we use transgenic method by using genes by using tree construction by RNA ISY in MIBCCD4 and CRISPR. But now we learn more about the which genes actually that we need to modify, or which gene that can we apply to modify the color. That's why we have to know the pathway of the pigment biosynthesis. As we know, in the left side is the pathway for carotenoid, and in the right side is the pathway for anthocyanin. So this one is the main pathway in marigolds because they mainly produce yellow, orange, and red. So that's why when we do conventional breeding, we may not get other color. So if we want to change the flower to red or purple, we have to activate the anthocyanin pathway in these plants. If they have the gene, maybe the gene is inactive. So we have to know how to activate the genes. But if this plant did not have the genes, we have to introduce others to these plants by transformation like that. So this is the important thing for us to know at the molecular level what happened in this. So next please. So we can learn from this plants from Oncidium. So if we see in the left side, Tagetes patula and Tagetes erecta, the vanilla one, and the patula is the red one. We like to make our flag color pure red and pure white. How to make that these flowers to be pure white and red? So from the patula, we only like to degrade the yellow color, then make the red more intense, right? And for the pure white color of erecta, the vanilla one, we only need to degrade the yellow, and we hope we will get the pure color. So we can read the arrangement of white and red marigold for the our national celebration on August 17. Now, so we learned from the Oncidium already published. This is from Taiwan also. So the wild type is yellow. This is the wild type. This is Oncidium. Then this is the transgenic one by using PSY, RNIE. So after the gene is expressed, when the PSI is expressed, PSI is key gene to synthesize the carotenoid. When the PSI is expressed, the transgene that we introduce to these plants will degrade the RNA. So because the RNA have been degraded, the biosynthesis of the carotenoid will be stopped. That's why they cannot produce the yellow color. So from this methods, we can try to do it now on marigolds by using the RNI PSY construct. Okay. Next, the other approach to modify the color is by using the genes from Chrysanthemum. So the white chrysanthemum, why the color is white? Because chrysanthemum white has the genes that when the genes is expressed, he will degrade the carotenoid. So every time the carotenoid is produced, it will be degraded. Every time produced, will be degraded. So we can see here when the yellow jimba is the name of Chrysanthemum varitis. When the name of the gene is CMCCD4A. When this gene have the mutation, the color will be back to yellow. But you get the point? So the Prof. Dewa, welcome. Nice. Prof. Chan, Prof. Dewa already come. So I continue. So for white chrysanthemum, why the color is white? Because they have the CCD4A. When this genes is expressed, the protein will degrade the carotenoid. So the color will be white. But when the genes is mutated, the color will be yellow again. Because if the genes have a mutation, they will not active. They will not active to degrade the carotenoid. So we also use this contract by using CCD4A for our marigolds. And we use the specific promoters, means that the genes is only expressed in the petal, not in all plants. Because if the genes is expressed in all plants, the plants will be smaller, because carotenoid is also important for the plants to protect the chlorophyll in the photosynthesis. That's why we don't want all carotenoid is disappear in the plants. We only want the carotenoid is disappear on the flower. That's why we use specific promoter. Specific promoters mean the genes will be only expressed at the certain place, or at certain time, or at certain age of the plants. So in our construct from Prof. Chan, in MYB CCD4A, MYB is the specific promoter for to be expressed on the flowers. Next. So far, we already tried to transform these genes to our marigolds. Actually, we already get the plants, but the efficiency of transformation is still low. So we need to do more to improve our techniques to get the more putative transformant and to be evaluated to at the morphological characters or carotenate compound and also later the color of the flowers because marigold is only need a short time to be flower so we can see the flower color in 3 months maybe. Next, this is the other picture when we already transform the plants with the genes, then we select the colors in the antibiotic medium. So the selection medium is to 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 separate the cell that already be introduced by the gene and the normal cell. The normal cell will die in the selection medium, while the cell that already have the gene that we introduce, they will stay alive in the selection medium with antibiotic. So we see here, most of the cell die because this medium contain antibiotic. While we found some cells still green because they already introduced by the genes that contain the antibiotic resistance. Next. Next, please. So the next project already going, still in progress. So we use the another approach to make to choose to make the precise decision that what gene that we can use to modify the flowers. So we do the transcriptomic analysis from these two species. Then next, we found some genes are highly expressed in this species. The intense colors mean that genes express higher in this species. Then these genes are involved in anthocyanin. Anthocyanin is the pigment that will make the red, pink, purple color. So we already know that Tagetes patula could be a source for the genes that can be construct to be introduced to Tagetes erecta. Next. So I do not give a conclusion because the research are still in progress. We full thank to Universitas Udayana. Prof. Dewa is our research team. Thank you, Prof. Dewa, and to Bali Province government through agricultural dinas pertanian ya. Then to Prof. Chan, Akademia Sinika to support our research. Next. This is our meeting with the professor, Prof. Dewa, and Kepala Dinas. Then next is okay. This is our team so far. Next, we will be also support by Professor Takagi Sensei. I hope so. We hope Dr. Wayan Koster, Bali Governor, will still support us, Prof. Dewa, because our dream is red and white for our national flag. Thank you very much. And please, if any other question for my presentation. Thank you.
Okay, give
Assalamualaikum
applause
warahmatullahi
for
wabarakatuh.
Dr. Dewi. Udewi, yeah. Okay, so very nice presentation about the marigold, especially for make the red and white, so make the like our flight. Okay, before we start for discussion, maybe Professor Dewa, maybe make some addition information or some. No, it's okay. Okay, okay. Thank you very much, Professor Dewa. Okay, yes. First, I would like to invite the student first for the question before another. Any question for the student? Okay, so please, what is your name and give the question for who, please, your times.
Thank, thank you for the opportunity. I'm Prila Kartika Manulang. I'm from Agrotechnology Magister student from Universitas Udayana. I want to ask for two questions for Professor Takagi and Professor Ming Sertan. For the first question for the Professor Takagi. Okay, thank you. Okay, for your presentation, I want to ask about which gen can regulate plant resistance to acid soil, and is it easier to us to lower the acidity or use the biotechnology for the engineered genes for the plant that resistant to the acid soil? And for Professor Ming Serchan, I want to ask about that we know that the genetically modified product for the regulation is still the healthy side for us human who consume it. How about your how can we know that the GMO is safe for us human to consume it? Thank you. Is it easier to us to lower the acidity soil or we engineer the
Good system. Oh, a good system for the screening, right? Okay, so we are selected the it's not only knockout. We use something specific gene silence system, and we finally we get some genes in arabidopsis. But arabidopsis and rice is very different. Monocot and the dicot. Okay, the first we have to study it in a rice. Okay, okay. Then so it's the answer is possible, but we still didn't do the inner rice. Okay. Yeah, but okay, answer is possible. You know, so because we use the actually we use a transcription factor. Okay, we modify the transcription factor. Transcription factor conservation is very high in among the plants. Okay, okay, so then we can easily isolate it as a homolog, ortholog in the rice. The rice we can transform. Okay, but another plant very difficult, you know. So it's impossible. But we have to first develop the some transformation system like a marigold, something like that, you know. The rice is we can do that. So, but at the same time, we have to overcome the problem with the GMO because we eat the rice. Okay? So the loss of the GMO is not direct eating, you know, just make oil or some the feed or something like that. Okay, that's also second problem whether they accept the GMO plant or some biotechnology. Something. Okay, but it's good question. Okay, I like that.
question is how do we deal with the GM? Yeah, I think that to be the scientist, that we have to educate the population, popular, common society. Yeah, so we have framed that how many them advertising advertise advantage, yeah, for the society. Yeah, so for example, that stress tolerance and increased years, and to fulfill the food. Yeah, so that is the advantage advertisement advantage. Yeah, so but we still have to manage the disadvantage. So about, for example, the pollen distribution and some other society issue. But I think that the major issue is the GM regulation is depend on the government, right? So if you can have the chance to talk to the government, then you should educate them. Yeah. So example that we always educate our government, say how good the GM is good, but why GM? Or for example, that why yeah, like a professor just asked me why I studied the orchid, because we did not eat the orchid, right? So when we modify the orchid color or orchid virus resistant or orchid disease resistant, that is the might be the first step to convince the people to believe the GM, right? The second might be when the food is less, might be the GM food might be can be acceptable. So I think that we have to convince the government and the people. Yeah, that is my. But I think that GM politics might be another issue, because that, for example, that why do you see that why the Europe did not protect, did not allow the GM? Do you know that? That is the politics, because their technique, their technology might be catch the level as compared to the American, because too many American import food into the Europe. So I think that is politics, not only for the safety. So okay, so one matter maybe have two sides, face, two face. You maybe think about that. Yeah. Okay. So if the Europe country, the scientists, uh, the result can be improved a lot. I believe that the GM will be allowed in the, like the Japan, right? Already, the GM, they are very stringent in the Japan, but now that they already have the blue color, rose, oaky, and even some, I think, the sweet potato or, but forget one, yeah, yeah, right. So our first step is to want to change to the horticulture. Yeah. So because I think the same technique we can use in the crops. So for example, the rice, yeah, rice transformation, we published that in 1991. We already created might be more than 50 cultivar, but we cannot use that. So I think that's my thinking. Yeah, thank you.
Yes, related with the question about the GMO, also in the chat, have the one question about this one for the Professor Takagi. I think already Prof Takagi try to answer related with the GMOs. Okay, please.
That's question that you gave me.
No, before, so you read in the.
Oh yes.
Okay.
the question in my talk in a chat. So how the acceptance of the GMO in Taiwan. Okay, so I'm a very beginner in living in Taiwan, so I asked the minister. So Professor Chan, he already part of them reply that. Just in Japan, it's stationed in a GMO. Okay, I can just inform you the stationed in GMO. Okay, GMO in Japan's government allow any GMO, if they pass the some regulation. Okay, and also the genome editing is not considered GMO. So if the product is made by the genome editing, you can sell it anywhere without permission. Okay. But in Japan, it's a GMO is kind of the something commercialized. Oh, I have heard GMO. I don't like to eat the gene or something like that. Everyone, you don't you don't want to eat the gene, DNA, or something strange. Strange. But the people didn't realize. So in some food, they say we don't use the GMO, even if there's no GMO plants. So in GMO, in Japan, only one gene is imported. All right. So there's a huge amount of the GMO plant is produced, but in commercially, people really surprised that only one gene is a Roundup high road resistance, and also second is a BT toxin. But we eat the GMO just only soybean as a nut or tofu. Okay, another food we don't eat it directly. If we consider the oil is not direct to the food. Okay, so there are lots of soybean that produce the oil, but we eat such oil. But I don't think it's not involved in GMO anymore. Okay, scientifically. But the soybean we eat that, tofu we eat that. But but the people they don't know that just only one gene is used in commercially in Japan. Yeah. And also the flower is the blue rose is the people accept it. Okay, so because it's not we don't eat. That's the station of the GMO in Japan. Okay, please.
Taiwan, we allowed the GM import from the United States or Canada. Yeah, we eat a lot of GM, although the people did not know that is GM. Okay, because we eat a lot of soy milk, tofu, and corn. I think might be more than 90% is the GM. Yeah. But maybe people did not know that. Although because our regulation is if the GM material is not over the 5% material, that you can be labeled as the non-GM. So and also when you cook the soybean as the soy milk, that will be not the GM. Yeah, like a soya oil is also not. Yeah. But unfortunately, we create a lot of GM crops, but already import to the mainland China. Yeah, through the unlegal way. Okay. Yeah, but our Taiwan government did not recognize that. So because we are very close, so just through the lead, then we can import that a lot of through the farmer and some tourists. So that now our GM still on the field trail for more than 10 years. Okay. So that's why I trying to change my material as the horticulture. Yeah. Sorry. Thank you.
I think Prof. Takagi still have the question from the chat.
Yeah,
So please
yes.
answer. Okay, thank you.
Yes. It's okay. Can hear. All right. So the question is which vector is most acceptable for the transformation? So we use the base of the BPI vector. but the vector is not so important. It's agrobacterium. Okay. Question is when we transform it to the plants by the agrobacterium, which vector is most suitable? Okay. So we use the basically BPI vector, and but vector is not so important. Its structure is same. It's a right board and left board. What is more important is the strain of the agrobacterium. Okay, that's most important. And the other genes, hmm? Modification. Modification is gene. Question is what is a better mute? For the modification of the genes, which is better, a mutation or genome editing? Okay. So the answer is if you have we have the mutant line, so we can use that. Right? So we don't need to make the CRISPR-Cas9 vector. But if we don't have a CRISPR-Cas9 is very useful. And also now the CRISPR-Cas9 is just a cassette vector. Just you put in a sequence that you want the target gene. That's easy, right? Okay. And the other question is what factor we should consider for the manipulation? Which factor means which genes? It's a very difficult question. Which gene? All right. So in my presentation I showed that the one just modified one gene we can modify. But it's a presentation is not um, is fact. But actually, even if the as a mutation in the plant mutation, just single gene, most of the plants do not show the phenotype because the plant has a lot of the redundant genes and the genome is so duplicate, So I used my way is some dominant repressor and a dominant gene silence system. So the just one gene can suppress many genes. That's our system development. This time I don't explain that, but we developed some overcome redundancy. So for example, marigold has also and also the chrysanthemum has a hexaploid. And also wheat is hexaploid, tobacco is a tetraploid. So just sometimes one gene knockout do not show any phenotype. In Arabidopsis, it has 98% of the genes is knockout, and also most of the transcription factor genes, there is 2,000 transcription factor is knockout, but only 10% show the phenotype, right? Because of the redundancy. So that's we have to think about some overcome such a redundancy. That's most important.
The term now for Dr. Dewi. So there are two questions. The first about how do will you compete for the government that what you do the result. Okay.
Thank you for the question. So, how will you convince the government that what you do and the result is safe and good for the environment and health? Yes, our government already have the regulation for the GMO. So, there is the law for GMO in Indonesia. We just follow the rule, right? So there is some steps before the GMO can be released to the public. There is the biosafety test, environment safety test, food safety, and feed safety. So we call it the uji keamanan lingkungan, keamanan pangan, keamanan pakan. Maybe Prof. Dewa can also inform us. So just follow the rule, I think. And so far, there is no proof that GMO give the negative effect. Mainly like for soybean, we already consume it for very long time, right? But I do not know, is there any research to evaluate its effect for our people? There is no information, like, or for our environment or our animal, our so just follow the rule, I think, to get the new what is it? The government to fulfill the government need for one new varieties of GMO.
That for the question is about
Black
the
jeans.
black
Yes,
jean.
but the
Okay.
black jeans, maybe not black jeans, but the jeans that can give the black color. So Prof. Chan maybe later can add the information, but I think for the black jeans, actually in flowers, if we saw the um, flower color, some are black. Actually, it may purple or deep purple. Deep purple. Deep purple is produced through anthocyanin pathway. So we can overexpress the certain genes mainly in the delphinidin pathway, right, Prof. Chan? In the delphinidin by using the what is that? Overexpression, overexpression in that pathway. So the color will be more purple or deep purple. But if we see there are some part of plants that black, like seed, the seed of marigold is black. So maybe there are the genes that involved in producing of the black color on the seed coat. That's why by using the transcriptomic analysis from this from the part of from many parts of the flowers, we may know what genes are exist in these plants and what chemical by using like HPLC we can analyze the compound exist in the seed. So by this information later it there is the genes in the seed. The genes is also exist in the flower. But why the flower is not black? Because the genes is not expressed in the flower. And how to modify the flower to be black? So we have to active these genes in the flowers. So maybe we use the specific promoters or to activate the genes in the flowers, or we edit some part of the genes, but because the genes is already exist, because the genes in all cells is similar, in the vegetative part of the plant is similar. The genes in root cell, in stem of the cell is similar, right? But certain genes are only expressed in certain parts, only in the cut seed, right? So now we may think how to express these genes in flower. What makes the genes is not expressed in the flowers? We can learn it by using biotechnology, by using transcriptome analysis. It's very interesting subject to be learned. Prof. Chan, could you add the question about the black color?
Red color. Okay. That is our secret, right? So if you want to know, please join us. Yeah. Yeah, because we have the flower specific promoter and to overexpression some genes. And also according to the Davis data, we already know some gene not yet not expression in the marigold flower, so that we can adapt from the other species that genes to that isocyanin pigment to go through another pathway. Yeah, just that I discussed with the Ibu. Just discuss with, and later then because the dark the black is almost like the dark purple, so then we can increase the pH more. We can use the pH modifier because the pH modifier then can that the color to be deeper, deep colors, and we already have candidates to do that, and also as like David already mentioned that why the medical seed containing the black color, black pigment, but why not expression in the flower? So that is another research topic that we can dig into, because that I think they may accumulate a lot of phenolic compounds, so that we can clone that phenolic compound synthesize gene from the seed, marigold seed, and also junction with the brown specific promoter to expression in the duct. So that our final goal is maybe we want to create a white color, pure white color, pure red color, and pure black. And also that white come up with the red, like the Indonesia flag, right. That is our purpose. But I think the more detail, please join us. Yeah.
It is give the chance for the join for this our research. Okay. Now maybe still have the time about 10 minutes for the question. If any question. Uh, so, before my Prof Dewa have comment. Prof Dewi, yeah. Already. Uh, okay. Yeah.
Oh, yeah. Thank you for the chairman for the chance for me. I introduce myself. I'm Iketut Swade, lecturer in this faculty, Faculty of Agriculture. Thank you for this chance. Many thanks for the very nice presentation. I have many many things that's very good for me. Yeah. I have a question. I address to Prof. Takagi. Yeah. Actually, in gene transfer, we construct plasmid, of course. There are some genes, for example, the insect gene, and also the selectable gene, as for example, agromycin or streptomycins. Yeah, that is selectable. Of course, the gene will also arrive in the genomic of the plants that we going to. Yeah. So the gene also join together in the plasmid or chromosome. So my question is how to eliminate the trait, I think. Yeah, because there is many people that don't know about that is afraid of the genes. Yeah. That is my first question. And my second question is to Prof. Chan. Yeah, Prof. Chan, thank you. Oh, oh, yeah, yeah, yeah.
The you can hear. The first question is we made transgene. It's a protein, the plasmid, and transform to the genome. Okay, so the question is how to eliminate the something, some light board or something like that, right? So it's very difficult. So it's elimination is difficult. That's the idea. Some genome editing is developed, you know. So but the but the most of the genes is something associated part. It does not function at all. So some GMO plant in United States. Oh, so it's a kind of they check the safety test, and then if they pass, they then take care about that. If the some associated part has encoded in some gene, it sometimes make some problem, but they don't have any encoded sequence. So they do not affect the some product or protein production, translation, something like that. Okay. But the first question is how to eliminate. It's very difficult to do that. Pardon me? Oh, now okay. Recently, the people who are against the GMO do not about the some harm, uh, some health because some they only people something. This is very curious. There's a large number of the GMO, but only the three type of the genes is on commercial. Only three. Okay, so the people who are against the GMO do not talk about the some dangerous or harmful because it's scientifically nonsense. So now the people they change the idea. If we use a GMO, there's some something the poor people and rich farmer is almost something distance is more elongated. So environmental something like that. They change the policy to the health to the another subject. Okay.
Yeah. Thank you. I work in club root for cabbage. Club root. Yeah, that is the disease. Uh, uh, the disease, the pathogen is Plasmodiopora brassicae. The club root is very high intensity in in the plantation for cabbage. Yeah, also brassicae, most of brassicae. Because that is very decrease the very hard decrease for the productivity because of the pathogen. And what do you think? Uh, how to make the plants resistant, for example, to the pathogen? This is very problem. Yeah, I think because you have many equipment that is very easy to do. Yeah, but here it's very difficult to do because of many constraints to do genetically work here, something like that.
I think.
Make a plant resistant, maybe using genetic work, genetic modification.
Yeah, I think it could be, yeah, right.
It can be.
Yeah, it can be. And resistant to the insect or a pathogen, I think is also easier. Depends on what the insect you want to protect.
Panjai.
Fungi.
Panjai.
Fungi. Yeah, I think it's it can do doable because that fungi it depends on what fungi you may face. You have to first of all you have divided which fungi is belong to what's the species the fungi you may face, and also for example that the yellow banana yellow crown virus is like fusarium fungi. Yeah. Fusarium also, Takaki Sensei also do many works for the fusarium. Yeah. So like right now already have the several plant gene can be resistant. So you may find a wild species may be more resistant to these fungi, and to find that, or you may using another one already used. So depend because I'm sorry I'm not familiar with these fungi. So I may see that reference. Then maybe some gene you can pick up. Yeah. I think a lot of the fungi already can be resistant. Yeah. And also, for example, we ever use the one very toxin gene, the plant toxin gene. We published in 2015. We only expression because sometimes we eat the fruit, right? So we can use the leaf specific, like a physical small subunit, only appear in a green color, or root specific promoter to drive that the toxin genes. So that because the fungi they will grow in the soil. So you can use sometimes you can use the root specific promoter to drive the toxin genes to kill that fixarin. Yeah. So I think it's doable. Yeah. We may discuss because I'm sorry I didn't have familiar with this fungus. Yeah. So in a even in a insect, now we also have like several candidate genes can be to be transformed, like the rice I forget the term. It's a small insect. Yeah, in rice. Yeah. Right, similar like that one. We also use in one genes to overexpression that in the leaf because they eat the leaf. So that we only expression in the we use the rubisco small subunit expression in the green color. So then the insect eat the leaf, they will die. But the toxin gene, the toxin protein will not appear in the seed. So they will be safe. That is another escape for the regulation of the GM.
More? Okay. Can we? Yeah, just one thing about Dr. Dewi about marigold. I just want to know you saw the purpose is flag, look like flag, so red and white. What is your purpose actually? Is it the red in one plant or the same plant? Because there is a flag look like in one flower is made of red and white and red. Okay, thank you.
Yes, that's biotechnology. We can imagine, we can make what we do not imagine before. So as we see, there is marigold with red and yellow in one flowers. I think we also can make red and white in one color. Otherwise, we can make also totally red and totally white. So depends, and there is the technique to do that. Could be.
I think you can create a new variety only the white, pure white, and another pure red. And might be another one is the red mixed white. Yeah. Because we already using a CCD4 is the carotenoid degrade enzyme. So they will, and also the only expression in the flower. We junction with the flower specific promoter. We already send to the dairy. So the CCD4 will degrade the carotenoid. So that means the red with the yellow yellow color will be degraded. So you will see the white, pure white, come with mixed with the red. Okay, so you can imagine that. And the yellow color, yellow original yellow, because you also using CCD4 to degrade carotenoid. So that's why we can obtain the pure white. Okay. Then the red color is our next step, I think, because if when we degrade the yellow color, we need to add this another anthocyanin, genes. So what gene we did not decide. It depends on the transcriptome data result. Yeah. We still need a DAWA to support the grant. Yeah, because that she have to give us the budget for to supporting. Yeah. Right. We welcome the every student to want to join us. Yeah. We have a high salary for the scholarship. Yeah. Yeah. Right. Right. We only need an interview by online interview and some application document to apply. So we welcome every student as the willing to join us. Yeah. We're not so open to ask the question for the student, just except the Takaki sensei. Okay. Don't worry about that.
Okay. So still have time for the student. Okay. Okay. If you any question for student, ada? Okay. Okay. If no, so the committee already told me the time is up. So I'm sorry for this one. I think it's very interesting topic today for the talking about the macro genetic and also biotechnology because make impossible to make a possible. Okay, this one is very important. Okay, give applause for all our lecturer today. It's very nice topic and thank you for much. Okay, so of course. After that, all of lecturer maybe want to take a picture and also for the Prof. Dewa, Dr. Dewi said. Okay. Okay, thank you very much. I have to bet for the committee for the continue for the agenda. Okay, thank you very much.
Okay, thank you very much to Mr. Putsudirta S. PhD for guiding the presentation as well as the discussion session. Ladies and gentlemen, we arrived at the end of the seminar and we would like to invite the speakers to receive a certificate of appreciation and would like to invite the Eco Technology Master Program Coordinator to deliver the appreciation certificate to the speakers. Kami please. Ibu Rindang dipersilakan untuk memberikan sertifikat. Nggih. Okay, we would like to invite all of the lecturers who are joined with us today for taking a photo together. Yeah. Ladies and gentlemen, I would like to announce to continue our event today. It will followed by lunch break, and then we can continue our own agendas. Thank you very much.